Okay, so now it's nighttime 10:04. So ready?
Yep.
Okay, I'll start. Okay, so good morning, afternoon, evening. Hola, hello, bonjour. Ni hao, konnichiwa, 안녕하세요. And hello all, and welcome and thank you for joining us the 24th seminar and Rice University Day, probably potentially Rice University Day 1, because next week we will have another Rice University professor hosting here. So we are all connected, I believe. Without connection, many do not function, including the web. That could be real spider web, or internet, or guitars, the instrument to play, and even trains. I mean, all the rails and so on. Of course, the most importantly, the human or all living world. And today's speakers are all connected as well. Jeff Tabor is my academic brother in Chris Voigt lab, and he kindly switched it with Chris Voigt because Chris is traveling right now. And Fangchao is a postdoc in Adam Arkin's lab. Adam was Chris Voigt's postdoc advisor. So Fangchao is my academic uncle, although he is my official mentee through the AICHE mentor-mentee program. Indeed, I do not need to mention the 6 degrees of separation, a concept that any two people on Earth are 6 or fewer acquaintance links apart. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Jeff Tabor. He is an associate professor of Rice University. Again, he's my academic brother. And interestingly, I am following his footsteps. For example, he received numerous awards, including NSF Career and ONR Young Investigator Award, to name just a couple. And I also got both of them. He's a real true pioneer in probiotic engineering, and I'm also doing similar research. And most importantly, one of the IGEM, first IGEM projects that basically about the photograph, about photograph using the E. coli cell, and then that basically excites entire IGEM community and synthetic biology that published in Nature, followed by another great cell paper about the edgy detection, and he is the leading author of that paper, and then he's a truly pioneer in the synthetic biology community. So I'm super honored today to introduce him, also host him today. And with that, please welcome Jeff, and the podium is all yours. Thank you so much.
Great. Thank you, Tae-suk, and thank you for inviting me. It's nice to be here with you and Fangchao and the young people watching. And thanks for that really nice introduction. I'm actually going to talk about my path through synthetic biology a bit and touch on those things. So, when I thought about talking to young people about where I've been and what's inspired me and the lessons I've learned, I sort of started at the beginning. And so when I was an undergraduate, I was at UT Austin, and I worked for an evolutionary biologist named Jim Bull. And Jim Bull studies bacteriophage evolution. And he had me do a really formative experiment there that really sort of set me down a path of synthetic biology. And it seems kind of trivial maybe, but it was really impactful to me. So he gave me a test tube with purified genomic DNA from bacterial virus, Phi-X174. And I electroporated it into E. coli So I put the DNA inside of E. coli cells and I plated them on an agar plate. And the next day I came back into the lab and I saw live virus growing on my plate, right? And so even though I was an undergraduate, it was a really profound experience that I had held the genome of an organism in my hand and that I had been able to generate that organism from its genome directly. And just the idea of what you could do with that capability, if you could rewrite the genome and rewrite the properties of that organism, was really impactful to me early on. And so Jim introduced me to another faculty member at UT Austin, Andy Ellington, who many of you know, I'm sure. And I joined Andy's lab as a PhD student in approximately 2001. And like many of you, I was very inspired at that time reading these two papers, the repress later and toggle switch paper that came out shortly before that. And this concept that you could sort of combine biological components together in order to program the behavior of a living cell, I think was an exciting embodiment of I think what I was so excited about in that phage experiment of this idea of engineering life at the DNA level. And so those are very inspirational ideas. And Andy and I went on to think about, Andy's an RNA technology guru. And Andy and I began to think about whether we could put RNA in genetic circuits. So this is just a schematic of the repressilator here on the left, which is made of 3 protein repressors. And we wondered, could you put a ribozyme that acts like a repressor against the messenger RNA of one of those proteins and begin to build genetic circuits that way? And potentially you could build larger circuits because of the programmability of RNA-RNA interactions and so on. And this was early, and I did work to try to implement RNA-based genetic circuits in the lab. But probably a more impactful thing that happened to me is that Andy got us involved in the 2004 MIT synthetic biology competition. So this was actually, it wasn't called IGEM at this time, but this was sort of IGEM 0. It was 5 schools, and we were one of them. And the charge that we were to accomplish was to just engineer basically the coolest genetic system we could think of, right? And so we did, and it was a really great community of people that sort of got together that fall and talked about what they had done, right? And so there was a Harvard team with George Church. You can see there was a Princeton team with Ron Weiss, who was at Princeton at the time. There's a Caltech team with Christina Smolke and Michael Elowitz. There was a UT Austin team. And we got together and talked about things. And what our team had done, as Tasek mentioned, was engineer bacteria to see light and take pictures. And so this was work that we did by collaborating with Chris Voigt and Anselm Boskaya. They had developed a light-sensitive bacterial gene regulatory pathway. And they had kindly given it to us because we wanted to sort of engineer bacteria to take pictures and process images for IGEM. And so we did that, and we were able to shine light on petri dishes and get them to take photographs, which was a big hit at IGEM. And then I went on to work with Chris as a postdoc. So I moved on to Chris's lab at UCSF. Actually our original goal for that IGEM project was not to get bacteria to take the pictures, but actually to trace the boundaries of objects within images, so to perform the image processing task of edge detection. And this required connecting our light sensor to some genetic circuits and a cell-cell communication system. So I worked on that for a couple more years in the Voigt lab. And that was also a lot of fun and a fairly big circuit for the time. And then in 2010, I moved to Rice to start my own group. And one of the reasons I moved to Rice was that the students were really great there. And I was able to quickly assemble a lab full of wonderful people. And this is sort of an earlier picture of the lab. And we continued along some of the lines of optogenetics and light sensing bacteria, but we also went in some new directions, particularly programming bacteria to diagnose and treat diseases. And so the work that we've been doing there has really focused in on what I think is a really powerful family of genetically encoded sensors, right? And so in almost any synthetic biology application that people think about, there's usually a cell sensing something in some way, right? And so if you want to engineer a cell to be an autonomous agent and to go and do something, it typically needs to perceive its own state or perceive the state of its environment. And so this family of pathways called bacterial two-component systems, they're the simplest signal transduction pathway architecture in nature. They're sort of highly engineerable. And because there's so many bacteria, and bacteria have evolved to thrive in so many niches on Earth, these sensors have evolved to sense an extremely wide range of stimuli, from light to metals, to small molecules, to macromolecules, and so on. And so we've been trying to sort of harness these things. In the lab, we built a lot more light sensors. So we had a red light sensor early on that we did that work with. We built other sensors based on two-component signaling systems for optogenetics. And we've sort of advanced what's possible with optogenetics by sort of doing really predictive control of gene expression signals over time. And even going in vivo and using light to manipulate the metabolism of the gut microbiome in order to study how gut microbiome metabolites can impact the host. And finally, as Tasek mentioned, yeah, we've also been looking at two-component systems from bacteria on the human body, or even outside the human body, to try to sense biomarkers in the body, to try to program bacteria to diagnose and eventually treat disease. And so I just had a great group of people in the lab led by Christina Daffler early on, where we identified a new two-component system from a bacteria that lives in the bottom of the ocean that senses this gut biomarker thiosulfate, which is linked to colon inflammation, and sort of ported it into gut E. coli, fed them to mice, collected them out the other end, and were able to detect if the mouse had intestinal inflammation using a flow cytometer and GFP. So we're really been pushing on this direction now in the lab, and the science it continues to be exciting. I think the thing I want to leave you all with is sort of two lessons that I've learned during my journey, right? Which is the first is that mentorship is critical to your career in science, right? And at each stage of my career, I had a wonderful mentor who not introduced me to exciting science, but also introduced me to exciting people, and also, of course, gave me great guidance for how to sort of go forward in the career. And so, yeah, these people have been really important to me, and it's cliche, but I wouldn't be here without the influence of those people. And beyond your mentors, I think a thing that has really come out from my experience is that it's the people that you're around that make the science worth doing, that make the science exciting, that enable you to do the science at all. It's really without the people that in the environment you're in, none of it would really be fun. None of it would really be successful, and none of it would really be worth it. And so I think to young people, when you're looking to go forward, look at the people. So, when you're deciding where you want to go, what lab you want to work in, look at your relationship with your mentor, and look at the people that you're going to be around, and really optimize for those parameters. And I think everything else will work itself out. So that's all I have to say, and I'm excited to hand it over back to Tasek and hear our next speaker's talk.
Oh my goodness! Wow, this is the amazing talk again. The inspiring insight. Also, yes. The people, not money and not infrastructure, are advancing science and technology. And that's why we rely on young people like today's speaker, main speaker, and some other people, I mean, in entire world. So thank you so much for, of course, I mean, summarizing nicely the history of your career, but that also covers some of the, I would say, the mainstream, the synthetic biology history. And I also always wondering, actually, who was the plate holding the person in your photo, bacterial photography, the picture? Is it you or?
That was me. Yeah.
Oh, that's you. Okay.
Yeah.
And then is it the squid, in the plate squid?
It was supposed to be an E. coli, but it looked like a squid because we drew it ourselves. Yeah.
I see. Okay.
Yeah.
My mystery actually solved now. And then also I noticed, also I love the Andy Ellington's photo, showing his always exciting kind of the speaking style. I mean, that I love the picture a lot.
That's
Okay.
exactly right. Yeah.
You picked the right picture, I guess. Okay, thank you so much. I mean, that's wonderful. Oh my God. All right. So now the main speaker of today with the longer introduction, Fangchao Song. He is a postdoctoral fellow at Lawrence Berkeley Rational Lab with the Adam Arkin. Again, the Adam Arkin is my sort of academic grandpa. Of course, that's why he is my academic uncle. He works on developing high throughput method to reveal bacterial interactions, profile microbial phenotypes in complex microbial communities. Before joining Berkeley, he completed his PhD in chemical engineering with Dr. Da-Cheng Ren at Syracuse University, where he works on understanding mechanism of bacterial biofilms and antibiotic tolerance. And personally, I also know Dr. Ren. Actually, we also long time ago served as a panelist in one of the NSF proposal panel, and also after that we also met multiple conference, I believe. And after Dr. Song's training in engineering, microbiology, genetics, and bioinformatics, basically he said to me that he would like to apply high throughput experiment and supercomputing to answer basic biological questions such as the physiology and dynamics of human microbiome. So today I probably need to call today microbiome day, or all kind of connection consider. We may say mentor-mentee day, or whatever we need to say. And also for your information, if this video or the seminar, somebody watching and then who happened to be recruiter, he's currently on the market. So before he join some university, please approach him and then give the interview opportunity to him. Otherwise, you will lose very, very talented rising star, and then you will be regretting in next decade. With that, I wanted to welcome again, thank the Dr. Song today, and then take it away. This is all yours now. Thank you so much.
Thank you. Thank you, Dato. I'm going to share my. Okay, great. So thank you, Tato, for the candid introduction. And I'm so glad to talk after Jeff. And it's my honor to present my research in the SynBYSS series. Today, I'm going to talk about the application of droplet microfluidics in microbiology and synthetic biology. And in the picture shown here, you will see like the bacteria actually grow in the drop. A little bit more detail about me. So as Tayso introduced, I was born in China and got my bachelor's in chemical engineering, where I synthesized and characterized these carboxylene dendrimers used as a stationary phase in the HPLCs. You can see this is a polymer. That's how I get interested in polymer. Then I decided to go to study polymer. I moved south to the Zhejiang University, and I start to design and model the mild and solid polymerization process for the biodegradable polymer, polylactic acid, used for the process scale-up. After that, I came to US, Syracuse, New York, to start my PhD. And at that time, not polymer, but something related, I started to study the bacterial responses to polymer surfaces, and which is called biofilm. Luckily, I was one of the first group of people who demonstrate bacteria can sense and respond to the surface stiffness like eukaryote cells, and identify a few potential gene candidates which are involved in this mechanical sensing. So as I worked with some model organism, so in the near the end of my PhD, I'm thinking, okay, either I will go to direction to be more molecular biology for the model system, or I will try to fight with the more realistic or real microbiome, because in the nature, all the microbiome are multi-species, are very diverse, and are very frequently mutated. So then I decided to go to later. So I decided to choose what I thought the highest throughput method, which one is the high throughput sequencing, another is the droplet microfluidics. So that's how I talked with Adam, and Adam was super supportive. And then I ended up to be a postdoc in his lab and start doing genetics, high throughput sequencing, and droplet microfluidics. I try to combine them together to decipher the complexity of complex microbial community. So how do you relate to synthetic biology? I want to start from this well-known key principle of synthetic biology, like we design, build, test, and learn. And design, build, test, and learn. And design, build, test, and learn. It's like endless circle, but help us to get the optimal candidate for our purpose. In each cycle, we have tons of strains we want to characterize, we want to build. So this needs some high throughput method. There are some examples of the high throughput experimentation in 2020s. Dr. Huiming Zhao presented the iBioFab in UIUC in the previous SynBYSS talk. And also there are another example in industry, Xamarin. They use lots of robots and automation to do synthetic biology. And yeah, they met some problem a few years ago, but I still think this pipeline is good. I also attended another talk from Huiming Zhao at JBay, and when he talked about the iBioFab, he made a very interesting analogy about the iBioFab. He mentioned he wants to make the iBioFab to lead one for discovery and also inspire the whole community to design better experimentation, just like the first ELIAC was invited in UIUC. It's coincident in 1952. After that, we all know after 70 years, how this first computer turned to be the personal computer and influence the whole world. And this is the new Mac Studio just released this week. And you can see how small they are. And it's much more powerful than the first computer. So I definitely believe the iBioFab can make one for discovery by their own. But Dr. Huiming Zhao didn't mention too much about, okay, how could they turn to be the next high throughput experimentation? So how the high throughput experimentation in like 70 years later in 2090s. Very likely it will out of our imagination because nobody will know what will happen. There are always amazing things happen there. But just based on the limited knowledge from now, I think first, it definitely not be like build hundreds of IBL fab. Like we need increase the density, but not the volume, right? So how to increase density? This is one choice. We can use droplet microfluidics, and this is how the droplet microfluidic chip looks like. If you are not familiar with that, it's all in a very small size, 1.6 inch. We can integrate multiple chips inside. They're all parallel. In each chip, we can generate from 6 to 10 millions of microdroplet per hour. If we consider each drop as a well in the well plate can host a characterization or test, it means like millions of tests, millions of well samples in one chip, and we can do like 26 in parallel. It's like hundreds of millions experiments we can set up in 1 hour. So it's definitely very high throughput. How about we just made it? Sadly, the current knowledge and techniques are not enough. I just briefly searched Web of Science yesterday and like just have Dr. McFluidy and the synthetic biology in all fields. This is how the paper looks like. It's like 25 per year currently. It's good, actually, it's increased, but it's still very few. So as a comparison, we can see if I type high throughput and high throughput method like and synthetic biology, it's like 250 per year currently. This is how synthetic biology looks like. We know synthetic biology is a very hot topic now, and it's like 7,500 papers in 2021. I have to mention, even there are only 25 papers here, and the most successful examples are all like single cell sequencing or DNA. One good example is what Dr. Kalin Plaza presented in previous SynBYSS series and the job thing. But about the, they are all like non-cultivation based. So for the cultivation based method, basically we have very few people about this. So there are lots of questions we need to answer. So that's what I'm working on in Arkin Lab. I try to integrate the culture-based droplet microfluidics to different modular, different detections, and different devices to manipulate the droplet, and try to build different modular to help the phenotype testing and the characterization. As I mentioned, there are lots of questions we need to answer, like how stable the droplet is in a diverse environment. How can we design smart polymers which can be a controllable material? They can form a shell and release the DNA as we need it, and how to make this whole process robust, reliable, and user-friendly. So this is definitely like to build the droplet microfluidic high throughput center or warehouse. Definitely is a system engineering project. It needs a collaboration efforts from multidiscipline. So today I'm going to give 3 small examples what I did, and just 3 small blocks to build a whole new droplet-based high throughput warehouse. So I'm going to start from the first one. I'm using the droplet to phenotype microbes in different conditions. So all the results I show today are unpublished. So this is the high throughput phenotyping, and we all know the significance. We have hundreds of thousands of diverse microbes or mutations we want to test in different conditions. So the traditional way, like the IBL fab, use a plate-based assay plus automation. They are normally very expensive and also consume lots of polymers, reagents, and often it's plagued by the cross-contamination if you do the well assay. Droplet microfluidic can avoid all the limitations. And this is the pipeline of the droplet method, very simple. So we pull all the samples together at the beginning. So if you have a transposon mutant library, and it's already put together, and or we can just get the sample, the soil microbiome or water microbiome extraction, they already mixed. And then we generate droplet by Poisson process, and we make a single cell in each drop, and we let them grow in the medium. After grow, we put them together. We sequence the T0 and T with spike in. We add the same spike in. So you will see like everything compared to spike in. We will know if they grow or not. So very simple process. So we did multiple characterization for this method. First, people always doubt, okay, if bacteria can grow well in drop. So this how I showed like their bacteria they grow very well in the drop, and this is the data. So assume I use 15 different very diverse bacteria in my test. Assume they can grow the same density as the overnight culture of E. coli. So this is the same density of overnight E. coli. How many times they need to increase in the drop? So you will see they all somehow around this number because they are different strains. They definitely have different like density, but they all grow well. Second is we were also concerned like how the sequencing-based method are quantitative enough, because it's normally considered as a like very quantified method. So to validate that, I make this 15 diverse strains together and dilute it. There are no droplet, no culture, just direct diluted. So we know exactly the ratios between the three samples. This is just a name. It doesn't mean that 10 to 4 cells. It's just a name, like to show the relative numbers. So then I sequence them each species and measure the growth. Sorry, not growth. Measure the difference of the number in different samples. We will see like the dot showing the truth. They should be on this line, and the result should the result is pretty good actually. They can give us pretty quantified numbers for different species for different strains. If you want to see the variation for different species, here it is, and we do have some variation, but roughly the results are pretty good. Then we validate the method. So the top figure shows we measured growth by the OD, the very traditional way, and in 2 medium. So all of them grow in R2A. 2 of them didn't grow very well in xylose. And from the drop-based method, this is the threshold, like 200 increase, and all of them grow very well in R2A, and 2 of them you will see below the threshold, which are exactly what we get from the OD. So this method is actually pretty accurate. Also, there are bonus actually. At the beginning, we try to phenotype the growth, but actually just by one more step, we can get some interaction information too. So as I mentioned, we mix all the strain together at the beginning. So we just save 1 ml and do the, I call it pooled culture or bulk culture. They just basically grow together in one medium. You can image actually they compete. Their interaction between them. This is different from the drop culture, and this is the result. Some of them didn't grow well. This is the threshold I set up, the 200, and they all like around the threshold. Only a few of them grow a lot. They definitely outcompete others by eating most of the nutrients. So then if we put the increase in the 2 methods, the drop method and the bulk method, it could give us some interesting interaction information. Most of them, like you will see, the red is drop culture and the black is bulk culture. They get decreased. It means like they were outcompeted in the bulk culture. And this one definitely eat more nutrients and grow faster and they outcompete others. The two strains didn't grow by themselves, actually grow very well in the co-culture. They are cross-feeding. So we are trying to identify the metabolite cross-feed the two strains, and actually multiple strains can cross-feed the two strains. So basically, this method works well, and there are lots of benefits and advantages by using the droplet-based method, like save time, save cost, use less plastic, use less reagent, tolerant to the cross-contamination. And it can be potentially for some unculturable bacteria because we start from mixture. We don't need to do the pre-isolation before we phenotype all the environmental strains. Then I'm going to talk about the second example. So, oh, by the way, we are now using this method to do a very large-scale phenotype and try to link the phenotype with genotype in the project I work on in Enigma, because we have a lot of novel isolations we want to characterize. So that's what we want to apply this method to currently. So I hope I can share the genotype phenotype relation results soon. So yeah, it will be very exciting. The second one is similar method, but instead of using the isolation, we are using mutations. We want to phenotype the phage resistance of thousands of mutations. The mutation we are using is called the RB106 library. It's a random barcoded transposon mutant library. So basically, it just like barcode each mutant in a traditional transposon mutant library. This method was developed by Adam Dusbar and Adam Arkin a few years ago. So by barcode them, we can get the information of the cell increase or decrease by just simply PCR that region, that barcode. So the basic method of this is they are put together, we put in the medium. In T0, they may have like equal abundance, and after they grow, the number 2 decrease, it means like they cannot grow well, and the increase one, like they can eat the nutrient or they can grow well. The fitness is high. Vivek in our lab use this method plus CRISPR-I and DAPSeq, which is another high throughput method developed by Vivek to study the phage resistance. So exactly as what I said, they pull the RB106 library together and add phage by different MOI and let them grow, see which one survive. He gets lots of very interesting results. And but there's one issue. Every time we end up with just a few like receptor genes, which can resist to the phage infection right away and start take all the nutrients. And we think there may be some other mutation which can resist the phage, but that don't have enough time to get recovered from the infection or cannot grow fast enough to get nutrition before it's all eaten by the fast growing, like the loss receptor mutations. So this inspired by one experiment I did in my PhD, like when we do the bacterial persistence after we use antibiotic treat them and place it on the plate. I believe many people have this experience. You will see like the colonies on the plate have different sizes. Someone like grow faster, someone grows slower. They may need a longer time to get recovery from the antibiotic resistance. So that's how we end up by using this droplet method. So we mix phage and bacteria in the chip and get them in separate drop and let them grow, as I showed before. And after that, we barcode, we PCR the barcode and sequence them. Then we know the cell numbers, and based on that, we will say, okay, which one can grow with phage, which one cannot. So I'm using E. coli, RB106 library, and studied the infection of phage N4 and T4. Here, I'm just going to give you an example about N4. So realistically, the result, this figure is what I draw when we discuss that idea. And so if we plot the thickness or growth of the bulk result with the droplet result, so we can imagine if they are exact same, they just follow the y equal to x line. But if that mutation get outcompeted in the bulk, but they can grow in their own room in the drop, they were in this zone because they have high fitness in drop. Otherwise, if they outcompete others in bulk, but in their own jobs, they cannot take nutrients from other room, they will be in this zone. So we expect we will have more stuff in this zone because we will have more like mutant which can resist in phage but cannot grow in the bulk. So it's true, like we have more dot here and lots of mutation like grow more in drop compared to the bulk, and we can see the signal very strong. And another thing we need to mention, actually, the overall trend actually agree with each other very well. And we see the difference due to the method and the cultivation time. The left showing the droplet-based method and the pooled method or the bulk method of the RB106. You will see like this is a drop method. All of them are bulk method. All these genes have very low fitness, but very high in the drop. It means actually there's something we missed in the bulk culture. And also we find some interesting phenomena, like if we grow in different time, we see different results. For example, this gene, like the fitness is not very high, but get higher in the second day. It means like they may grow slower or get recovered slower than others. And we may need longer time to see them. We do need to validate the method. And I plot the results like the T4, N4 together. So one way to validate that if we can get all the targets we already know or we already see in the bulk RB106 validate. So the result is yes, we do identify all the targets we get from the bulk assay from our drop assay. Also, we have more targets found in the drop assay. I'm going to give you a few examples, and we validate them by the phage plaque assay and complement the gene, and the phenotype also get complement. So here's the wild type with the empty vector we use for the complementation. And then this is the mutant with the empty vector. You will see like the resistance different. And this is more the bigger difference. And then after we complement the gene, like the phenotype get restored. So all these genes are new genes which we have no idea about how they could be related to the N4 phage infection. So for example, I'm going to talk about this and this in the next slide. And for these two genes, just like totally uncorrected gene, we have no idea what the function they are. But for the CHB gene cluster, CHBC is the inner membrane proteins, and the VEG, AZ, and H and intermembrane proteins, which are the valine exporter. So I'm trying to figure out the mechanism, how the two can be involved in the phage 4 infection. The third example is an interaction. So I'm going to just talk a little bit about that. Yeah, because currently we are working on like characterize this method because we do have lots of versions to make it work better. And in the middle of the disclosure, so I'm going to just release some early version of the pipeline we did before the pandemic. So the total idea is like this: we want to get the interaction experimentally, microbial interaction in a complex microbial community. Let's say like more than 30 members. It's better to be 100 or 1,000, like the real natural community. So there are two methods normally we do. First, isolate all the string and pairwise them. So assume we are only testing the pairwise interaction. But we all know we cannot isolate all the isolates from the environment sample. So alternative way is like do the dilution to extinction, and then we will estimate the interaction from that. Either we have all the isolation, we do the pairwise mixture, or we do the dilution to extinction. They need lots of experiments. For example, this one, if we have all the isolation, we need probably do the 10 to 7 experiment to do 3 replicates for all the pairwise interaction in a 1,000 species community. And if we do the dilution method, it probably needs like 26,000 plates and need like a liquid handler work for 5 days, just fill this plate. So 5 days work by liquid handler is not a big problem, but this plate, I don't think anyone wants to. But for the drop method, because they can generate small droplets, like million droplets in 1 hour. So basically we just need 1 hour and a small tube to hold all the droplets. This is how the pipeline works. So we start from mixture of different species. We generate droplets, use the droplet chip by a Poisson process, and to make the maximum of 2 strings. It's definitely a Poisson process and needs some statistical analysis. We do that and we optimize that, and we proved we proved this process works by mathematical modeling, but I'm not going to talk about too much math in this presentation. So to simplify that, I just like use two bacteria in each drop to tell you the concept. And then after that, they either grow or not grow in the medium with their partner. On the other side, we need to have a barcode drop. So I start from oligo with a unique barcode. I use the same way to generate like single oligos in each drop and do drop with polymerase and primers and PCR each drop by droplet PCR. Then like the unique barcode oligo can get amplified in each drop. Then I will fuse this culture drop, this barcode drop, with another drop, including the polymerase and reverse primer. And this barcode primer will serve as a forward primer. And let's say buffer using this kind of like merger. You will see these 3 drop merger to merge them together. Then I do droplet PCR to amplify the 16S. Then I put them together, sequence them, and by separate different barcode, I will know, okay, they are together, if how well they grow. And this is the result. Overall, actually, it looks pretty good, but there are many problems I didn't show here. First is this only 5 member community. So the interaction from the 5 members. The reason I only do this is it's hard to increase more members in the community. So because the merging is not that stable, not user-friendly, and also there are some error in the like barcode oligo, single barcode oligo droplet PCR during the PCR, because I only filter the unique barcode in each drop. So after lots of replicates, I can fairly get maybe 20 to 50 replicates for each of them. And they have like a little, the big variation I didn't show here. This is just the average. So it means we need a better design to increase to make up possible to characterize like bigger communities. So if only have 5 members, we don't need drop, we just make them. That's what I'm working on with the V2. Actually worked pretty well so far. So we're characterizing that, and we hope we can deliver more on the way forward, and hopefully it can work for very well for very complex microbial communities. So that's all the three story I want to share. That's just a very small blogs on the way to build the big droplet-based high throughput warehouse or foundry. In the end, I'm going to share some of the perspective, like what I mentioned. The long-term goal, actually, I really hope we can have something like the iBioFab using droplet microfluidics, which we can quickly respond from any like synthetic biology or biology question and put all the modular we have together to make it work and integrate with the data storage management and the supercomputing like the AI to like deal with the big data generated from method, and then have a think tank to design new method, make new chips to increase the ability of the high throughput center. So as I mentioned, like our knowledge is not enough so far to build this center. So hopefully when the publication reach to 250 per year, like the high throughput plus synthetic biology, I will check one more time to see if something like this happen, if we can have a robust, user-friendly droplet microfluidic high throughput center, just like the iBioFab, built in either academia or industry. So this is something I want to say. This is one more thing. Actually, it's not necessary to build this center to make big impact. Even some steps, some blocks, some new techniques or knowledges in the droplet microfluidic can make big impact. There are two, probably the most famous examples, like using droplet microfluidic to do like droplet digital PCR and single cell analysis. Yeah, this method developed in Lawrence Livermore, and after they sold to Bio-Rad, one VP from the team go to found another one called 10X. Yeah, so another example, as I mentioned, Dr. Kellen Plaza have developed this drop thing to synthesize DNA. This is another example, and by its own, they can make huge impact. So that's what I'm thinking. So, and what I want to do is I want to build small blocks, focus on some small problem. I hope it can make big impact like this example I put here. At the same time, I want to put all the knowledge and technique together. Eventually, I hope I can build this kind of like reliable user-friendly center in the future to help with powering the whole key principle of synthetic biology. So in the end, I want to share this. The quote is by Dr. Cindybrenner. Like progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. All the work I did today are in Arkin Lab, and all the people in Arkin Lab are super supportive, and I really like the environment, and Adam is an awesome mentor, and I love him. And also lists all the people who are involved in the individual project I just mentioned. In particular, I want to say like Xun Liu is a world undergrad in Berkeley, and now is applying for medical school. Shuwen Ho is a research assistant here, and she is a student in biochemistry, and she's going to apply for grad school in the end of this year. William Chrisman is a brilliant PhD student in the Department of Statistics, and he built a model of the droplet microfluidic process and optimize the parameters we are going to use and prove this method will work mathematically. And he is going to graduate this year, and I think he's going to look for industry job. And I will also thank my collaborator, Adam Batid in UCSF, and the team in his group, Jiang Peng, Serial, Le Qian. I really enjoy talking with all the people in his group. Yeah, I didn't mention others like Celine and Sarah Peng here. And my funding is from DOE. It's the project called Enigma. Yeah, Tyso just mentioned I'm on job market, and if you have any question or if you have any possible opportunities and collaboration, please reach out. Thank you.
Okay, wonderful, wonderful. Fantastic talk. I have my question, but I already see some question. But before, basically ask this question, Jeff. Do you have question because you need to go soon, right? Or
Yeah,
comment?
thank you. Yeah, thanks. So that was a really cool talk, Fangchao. Very nice method and applications. I was wondering, those genes you found for N4 phage resistance, have you tested them to see if they give resistance to any other phages other than N4?
Great. Recently, we just test N4 T4. We want to like expand the phage, not working for T4. No, because T4 is like more lactic. So we find very few targets for T4 because most of them just get killed very quickly. So yeah, we are going to test more, right?
Thanks. Good luck.
Okay, thank you so much. And then yeah, see you later, somewhere. And also I have question from Caroline. Thank you for the excellent presentation. This is very exciting for the future of synthetic biology. I completely agree with you. Do you think there are any capabilities that need development in droplet microfluidics that will make big change in the field? This is kind of similar, one of the similar questions I have myself. So can you comment on that one, Fangchao?
Oh yeah, sure. Yeah, I think there are a few. Yeah, I think currently, not a few, many actually. So one example is, I talk about both cultivation-based method and non-cultivation-based method. In cultivation-based method, first thing we need to know, like if bacteria grow similarly in drop as they grow in like flask or plate or not. We assume they are in most, but actually we don't know. Even they grow in well placed, sometimes it's different from like big flask. For example, that's how people find biofilm. Like in big flask, they have very low surface to volume ratio. So if some physiology of attached cells like biofilm different from the bulk, it's just very, very few portions. They won't influence the overall phenotype. But if we go into the well plate, we increase the surface to volume ratio. We go to droplet, we increase further. So they have more surface, maybe it will be like biofilm. They like to form biofilm in that way. So the one thing. Another thing is like, we want to control this well because there are some leaking through the water oil interface. So it would be better to have, like I mentioned, smart polymer, which can form a solid interface there when we culture them, but release the cells and degrade this when we need the cell DNA from them. Then it will solve this problem, another material science problem. And also there are another manipulation problem, because you will see all the successful methods so far are only using droplet generation. Very seldom they are using merging or sorting or something, because they really need the experience to operate them. So so how to design a user-friendly sorter or merger or other thing, also others can be easily used by the broad audience. It will be one thing. And also like how we connect this droplet microfluidic with, I already do something with sequencing, but like with microscope with mass spec to detect the metabolites. There are lots of questions we need to answer, and each of them will have a very big impact in the application of droplet microfluidic in synthetic biology.
Wonderful. Actually, I have another question from Justin. This is a very interesting question because I also wanted to ask this type of question. So how could the high throughput droplets be used to investigate complex large community interactions, such as, human gut microbiome, and how could you increase the number of microbes that can fit in each droplet? And another question is, can the droplet be made aerobic and anaerobic?
Right. So I kind of already mentioned a little bit how this method can investigate the interaction using the pipeline. So for the second question, like how to fit more cells, as I mentioned, it's Poisson. Like we can change the Poisson process. We can change the density of the mixture of the microbe at the beginning. Then it's like, and also we can change the volume of droplet. It's like a mathematical problem. Like in the big bulk, we take this volume each time, how many cells we can get from each sampling, right? So if we have a high density, we get more cells. If we have low density, we get either 0 or 1. That's how we control. So we can definitely fit more. It also depends on like the volume we choose, how big the drop is. So normally I'm using the drop size between 20 micrometer diameter to 150 micrometer diameter. So of course we can fit more if it's bigger, right? So, but when we make it bigger, there's another issue I want to mention in front ahead is we need to consider stability. That's why I think we need design smart polymer, but not rely on the oil-water interface, because if it's bigger, we cannot culture in the high temperature or for longer time. It's not that stable. The smaller is stable. Normally, yeah, it is. So that's another consideration. So regarding the anaerobic and aerobic, it's a very good question, and it can do both, but we do have different issues in both cases. Actually, what I met in most of the experiment I did is aerobic. So I make a very thin layer and shake it because it's only 1 mil, right? So shake it a little bit, just like 80 RPM. And I have a oxygen sensitive strain. I can test, I can produce proteins in oxygen, and it's totally fine. And they can have enough oxygen to do that. But in that case, we need to avoid like as much evaporation as possible, because small volumes, they have evaporation issue, right? So so in this case, I prefer anaerobic. We just close the tube, put it in, and everything down. But we met another problem in anaerobic because in some of the cases it produce gas. I'm still characterizing this. In fact, yeah, because it's small volume. If they didn't release the gas very fast, it's fine. It won't influence the stability. If we release the gas very fast, it can like destroy the shape of the drop. That's another reason I think we need a smart polymer, and we need a polymer interface to like confine them. Then we don't need to worry about the stability. And then after we culture them, we release the cells by like degrade the polymer. Yeah.
Okay. We have passed, but I want to quickly ask one tough question, challenging question, because you mentioned earlier. So let's say 70 years of later, actually I envision many labs or do it yourself community may have automation systems to the SynBYSS experiment. That's kind of like I'm thinking right now. So could you comment on this point in terms of biosecurity, safety, and challenges and promises next 70 years? That's a big question, but I just want to hear from you.
You mean in terms of the current pipeline or the whole, like every high throughput
next many decades? I mean, so yeah, high throughput, and then that will allow even some people interested in synthetic biology just do it because by buying the cheap equipment, automation, and then that will create potential, but at the same time some risk in terms of security and the safety.
In terms of security and safety, I think compared with current, first I have to say the current automation, the robot plus plate assay is still the best choice because we know much more about that. It's more reliable, even it have lots of problems like have lots of waste and have some contamination issue. But I think the droplet method will be better in the future. The reason is I already present some of them, but I think the only thing hinder the application of droplet fluid is not that reliable or user friendly so far. But I think the progress in either chip development or technique development can make it better, definitely can make it better. So for the security issue, I kind of thought some like insecure like scenario in this case, right? So yeah, the only thing I think, the only concern is how we can make it reliable and robust. This is the thing. So actually, I would have thought like make each like droplet mass spec, droplet sequence, or droplet sorting merging as a modular. Then we can place it as a Lego added together. But then I realized, okay, in 40 micrometer scale, it's really hard to align them together. So it's an engineering problem. But I think someone can solve the problem in the future. Even I don't know how to solve currently, but yeah, if it gets solved, yeah, we can make some module and like assemble them like a Lego by ourselves. Yeah, it's challenging, but I don't think it's unachievable. So
Sure.
Yeah. So
the reason I ask the question is, as a scientist and engineer, we have responsibility for what we're doing, for the benefit of or safety of the public. So that's why I'm asking that question. And as a rising star, and also considering the professor job, I mean, you probably want to continue to think about those kind of things, because not only we're thinking about scientific advance, but also thinking about societal impact. Because I mean, there are some unintended consequence what we're doing based on what we're doing because of what we're doing. So that's the important question we need to ask, basically. Okay. All right.
Oh,
so well, of course that's tough question. I mean, even my colleague cannot answer some of the question I always ask. So don't worry about that. Okay. So thank all for joining and staying today. We will meet again next week on March 17th, Thursday, the same time, the same Zoom link, and we will have Professor Matt Bennett at Rice University again. So Rice University Day 2, and we'll also have Dr. J. Armando Clases Molano at the Mike Smansky Lab. Mike is also my academic brother. So I would say we are all connected really. And as usual, the follow informal chat will occur without recording. And so please stay here if you are interested in chatting with us. And I will promote you to panelists who can speak and show your handsome and pretty face. And I will stop recording now. Let me stop.